Hi, I'm Cynthia Tifft. I'm the Deputy Clinical Director of the National Human Genome Research Institute. The Undiagnosed Diseases Program, and now Network, is a group of clinical sites whose responsibility it is to evaluate patients who have been in search of a diagnosis for a long time and have been unsuccessful in arriving at one. And at the National Institutes of Health, we see these patients. We put them through a week-long inpatient admission to carefully and deeply phenotype them. And then we do next-generation sequencing to try and determine if they have as I tell families, a more unusual presentation of a more common disease that's been described before, or a completely new disease that no one's ever seen before, or perhaps a combination of diseases, maybe more than one disease, that makes it look unusual in its presentation. There is, if they Google Undiagnosed Diseases Network, the first thing they will see that comes up on their Google search is the application to apply. The parents apply online. They say what the situation is with the child or with themselves. This also involves adults. And they upload a referral letter from their primary care provider, pediatrician, neurologist, nurse practitioner, and then they are sent, or their application, the request for their application, is sent to the closest geographic site in the country, and that site then contacts the family, says, please send me, or let us help you gather all of the medical records on your child or yourself if you're the adult. We review those, we decide if we think that we have an idea or that there's something that we might have to offer the family, and then we bring them to one of the clinical sites and evaluate them. We have 7 clinical sites, including the National Institutes of Health. There are 2 sequencing cores that do all the next generation sequencing, whole exome sequencing, whole genome sequencing. We have a model organisms core facility that basically takes potential new diseases, and it's not enough to just associate a disease gene with a particular patient. You actually have to prove that it's that gene that causes that disease. And the way you do that, if there are no other patients with the same disease, is to make an animal model. And the model organisms core would then make an animal model, look at the animal, see if it sort of matches the patient. And that's another way we can say, yes, this condition, this gene causes this condition. Roughly speaking, in the pediatric, I can only speak for the pediatric patients, those are the ones I see most. I would say maybe now 20% of the time we'll say, well, maybe it's this, they haven't tested for this yet, they should test for this one thing, and if it turns out not to be that disease, then we'll take them. We call it, so we will reconsider their application after they've done that testing. That happens about 20% of the time. About 20% of the time, they've had such a thorough workup, or their findings are so nonspecific that we really don't think we can add anything. We take, in the pediatric side, probably 30 to 35% of the patients that apply. So the odds are not so against you that we will see you in the program. I would say when we started the program in 2008, whole DNA sequencing, whole exome sequencing was not that widely available clinically. Now it is available at multiple laboratories clinically. So the patients that we're now seeing are patients who've already been through that process. They've been through a clinical exome, which basically scans the genetic information of the patient that says, do they have a genetic change in any disease that's already known? Not the unknown ones, but anything that's already known. So by the time they get to us, they've already exhausted that. It's not a known disease. And so it's a little harder now to try and figure it out. Many of them turn out to be new diseases, actually.